Hi, I'm Dr. Jack Cush, Executive Editor of RheumNow.com, and this is the RheumNow ACR 2016 Week in Review. The ACR meeting ended 2 days ago in Washington, D.C., and we're back into the usual swing of things. And at the end of the week, I wanted to review for you the most important things that happened this week on our website. If you hadn't noticed, this week on our website, we had tremendously expanded coverage of the meeting. Thanks to a grant from Lilly, we actually were able to get a number of reporters involved and cover the activities in a highly significant way. I want to thank the hard work of Dr. Cassie Calabrese from Cleveland, Dr. Arthur Lau from Toronto, Dr. Rachel Tate Houston from Dallas, Texas, and Dr. Olga Petrina from New York City for being busy bees and covering posters, sessions, plenary sessions. They were tweeting, they were recording, they were getting it all out there. If you'd like to get something from the meeting and you weren't there, you should go to our website to ACR16.RheumNow.com, and you can see day 1, day 2, day 3, and day 4 coverage of all that happened and all that we found that was significant. The website is full of news articles and feature articles and videos on many of the topics that were covered at the meeting. In today's session, I'm going to review some of what I thought were the bigger events and also some of the things that we didn't cover. But I'm going to start with a few things we did cover that I thought were big splash sorts of reports that are going to change practice. This begins with JACTA. This is a report of tocilizumab in giant cell arteritis. These were the week 52 results of people with GCA who received either placebo or tocilizumab. Everybody received high doses of steroids. It was 250 patients. It was reported by John Stone in a plenary session. Again, one of the largest studies thus far. But the bottom line was that tocilizumab was steroid sparing. Patients were taking, in the end, significantly less steroids compared to those who didn't get tocilizumab. And tocilizumab, at the end of the year, was able to induce remission in more than half the patients, which was significantly, like 5 times, 4 times greater than that seen in the placebo control. This is a game changer. This will be used as a registration trial. I would assume sometime next year that this will be approved. Getting it used in your patients at this point is going to be hard because it's not yet FDA approved, and it would be off-label use, but you should look forward to this in the future. I think it will change the way we treat this and other forms of vasculitis. The next big splash event was the results of the precision study. Widely reported. If you haven't heard the results, it was reported by Elaine Husney and Dan Solomon and a bunch of other investigators. The project was headed by Steve Nissen from the Cleveland Clinic. It was a 24,000 patient trial, took 11 years to complete. It was ibuprofen versus naproxen versus celecoxib in patients with osteoarthritis or rheumatoid arthritis. Osteoarthritis made the biggest bunch here, with 90% of the population. In the end, it was shown that celecoxib was no worse than, was therefore not inferior to ibuprofen and celecoxib when it came to cardiovascular outcomes. In fact, it was in some analyses significantly better, with up to 20% protection against cardiovascular events compared to ibuprofen and naproxen. The other big ticket headlines from this was number one, naproxen didn't do as well. In most other studies that the COX-2s were compared to way back in the Vioxx era, naproxen certainly looked to be cardioprotective by comparison with ibuprofen and diclofenac. And many of us have been using naproxen because of that data. Well, in this trial, naproxen didn't look any better. In fact, it may have looked worse. And I don't know if that's because they selected for patients with cardiovascular disease or at high risk for cardiovascular disease in the study. But those were the results. The other thing was that celecoxib was actually better at avoiding GI events and also renal toxicity, which was a bit of a surprise. Look for those reports. They're actually in New England Journal at this time, and I think there'll certainly be a lot of talk about the precision study. Osteoporosis made a big splash at this meeting. A lot of reports, and I'm not going to get into them. You look at the website, you'll see things on denosumab and a lot of anabolic therapies. Actually, Peter Nash said something very interesting in one of our meetings, that was that the days of bisphosphonates are closing down, and it's time for anabolic therapy or therapies that build up bone mass to probably take over. And I think there is certainly a trend in that area. Abaloparatide, we reported on in RheumNow before, and has been in the New England Journal and other journals, has been shown to be beneficial. In this particular study that was done at the ACR, it was very beneficial compared to placebo in reducing fracture rates in postmenopausal osteoporosis patients. You should look for that report. It's also been in the literature. The report here comes from Kosman and co-workers. The FRAME study looked at the anti-sclerostin drug, romosozumab, in women who have postmenopausal osteoporosis. This is a 24-month trial. I believe at the end of 12 months, romosozumab was significantly greater than those who got placebo, and actually resulted in about a 70% reduction in insufficiency fractures. Patients then were followed up with denosumab going forward, and you'll have to look for those results in other reports. There was a structure study of romosozumab in also in women with the same kind of reports that comes from Langdal and colleagues. And then, well, there just were many, and I really, I think I could spend a whole hour talking about those results, and maybe we should in the future. To cover a few more things from the meeting, a novel new compound called voclosporin. This is a new calcineurin inhibitor that's been in the news. It has had some phase 2 trials that look good. They had a phase 3 trial in lupus nephritis that was reported. This is called the AURA study that had 265 patients that looked at lupus patients who were taking mycophenolate and low-dose steroids, and then either gave them placebo, low-dose, or high-dose voclosporin. 65% of these patients had class 3 and 4 nephritis. 80% of the population was either Caucasians or Asians, and the primary endpoint was complete remission or complete response on low dose, and that endpoint was met significantly so. The other major thing from the study was that there was a rapidity of response certainly at the end of the study at 24 weeks. Over 50% of patients on voclosporin, and it didn't matter whether you were on the low dose or the high dose, had achieved their endpoint, whereas that was only achieved by less than 30% in those that are on placebo. But again, it was the rapidity of response, with response being seen between 6 and 12 weeks on voclosporin. Toxicities, and not surprising, a lupus population on immunosuppressants and steroids, a lot of infections, GI problems. Serious adverse events were about 25% with voclosporin, about 16% with controls. There were deaths either from infection, cardiac, or pulmonary causes, but that death rate was similar to that seen with the other large recent lupus nephritis trials, the ALMS trial, the ALLURE trial, and the BELONG trial, also showing death rate of 3 to 4%. So again, this could very well be a new drug to deal with in the future. At this meeting, there wasn't a lot of splash about anifrolumab, the interferon alpha inhibitor, which was a big ticket item at the EULAR meeting. There were some reports, but nothing new from that data set. There was a lot on the use of rituximab in lupus, and I'll remind you that rituximab in lupus has failed in clinical trials and is not currently approved. But nonetheless, a group of investigators at this meeting, I looked at a few and focused in on 2 British studies that basically showed in the BILAG-BR cohort and in a single center retrospective cohort, over 300 patients who received open label rituximab, really with good results. Of course, a lot of these patients have been on other therapies and not been doing well. In the open label trial with 79 patients, the response to rituximab was 67% for arthritis, 60% for cytopenias, 43% for vasculitis, and 27% for nephritis. Not as high as you would like for the most difficult aspect to treat. But again, in looking at these patients, especially in retrospective studies, it does look like they do better in open label use in the hands of rheumatologists. But in clinical trials where the rules get tighter and the outcomes are much more restrictive using BILAG outcomes, it's not yet approved. I don't use rituximab in lupus except for cytopenias, where I think it works really, really well, and unless I get into a situation where I'm frustrated. I find it interesting that many rheumatologists like to use rituximab in lupus, but yet are afraid to use it in rheumatoid arthritis, where it has a 1 in 30,000 chance of causing PML, whereas in lupus it probably has a 10 to, I don't know, almost 100-fold higher risk of PML. So again, there are some problems here with the use of the drug. They're rare problems, but nonetheless, I think we have to be smart about who gets it and in what situation. A nice study that was brought to my attention by Artie Cavanaugh was this report about how people store their drug. And this was a study of golimumab patients who were given a drug that actually had a temperature sensor attached to the prescription. And they monitored patients and what happened to their drug when they brought it home. Turns out almost nobody kept the temperature and the drug in the right optimal storage conditions, that there was many where it was the temperature was too low, or some there was too high. The idea is that patients may not be doing a very good job of storing these drugs. What is not known from the study, however, is did it affect the outcome clinically? So we'll need follow-up data from that particular group led by Dijong and colleagues. There were two important studies presented at this meeting about JIA, specifically polyarticular JIA, where two drugs, certolizumab and abatacept, were seeking approval for use in polyarticular JIA. It turns out that what's novel about these studies is that aside from getting the usual efficacy and safety outcomes, the primary endpoint, as mandated by the FDA, was PK. That they wanted to show that dosing in very small children and very large children of different ages could be smartly done using the adult dosing target as a starting point. They were able to define the doses for the different groups with both abatacept and with certolizumab, and did achieve their target trough levels. So you'll probably see these two drugs being approved next year after the FDA fully reviews these data from the two manufacturers. Biosimilars continue to be a big talk. Their biosimilar companies are exhibiting at the meeting. They didn't have biosimilar reps, though. They look like the other reps, as best I could tell. But nonetheless, I think that the big study at this meeting was the North Switch study. It was previously reported at the GI meetings and has gotten a lot of talk. If you're not familiar with this, Norway was one of the first companies to adopt infliximab, a biosimilar called CTP13, over there called Remsima, in other places called Inflectra. This drug is currently FDA approved and is due to be launched here in the United States sometime in December, early December. But the North Switch study, the terms of that adoption was that they received the drug at a 70% discount, and in return, I don't think there was actually a quid pro quo here. I think actually that it was asked that they do a study, and they did, where they took patients with many different diseases: RA, PSA, SPA, IBD, and obviously psoriasis, who were on infliximab doing well, and then they randomized over 500 patients to either receive continued infliximab or the biosimilar CTP13. And in the end, when they looked at the outcomes a year later, that the idea was, did they have any worsening, significant worsening for these many different conditions based on standard measures of efficacy in those conditions? And they showed as a overall that there was no there was no inferiority displayed, so that the biosimilar was non-inferior, and that there were no new safety signals. But when you comb down and look at the individual diseases, whether it be IBD or ankylosing spondylitis, there's quite a variability in how much the therapy favored one drug or another, and it didn't always favor the originator drug. So, or certainly didn't always favor the biosimilar. So the idea is that we need larger trials, maybe observational trials, once these drugs get out into the main, into the public use, to see if they do in fact yield the same results. Again, these drugs get approved with only 1 or 2 clinical trials to prove their efficacy, and then the efficacy of that one study is extrapolated to many different disorders across the same spectrum that the originator drug is approved for. So that, again, when the North Switch study is a pivotal trial that will be used as evidence for why we should be using more biosimilars in the future. The Scleroderma Scott trial, this was a head-to-head trial of a 54 months trial of either receiving monthly cyclophosphamide or myeloablative stem cell therapy in patients who have diffuse systemic sclerosis. In the end, you know, event-free survival and overall survival was greatest in those who received stem cell therapy, suggesting that this may very well be an important future therapy for us. Again, more is needed, but the results are fairly impressive. There was a nice report by Kevin Winthrop, an infectious disease physician who plays a lot in the rheumatology arena and follows our data very well. Kevin's report looked at the incidence of shingles in patients receiving baricitinib for rheumatoid arthritis. In his report, he showed that the event rate for shingles was roughly around 3 to 3.5 events per 100 patient years. That's a notch below what was seen previously in the tofacitinib studies, where it was around 4 to 4.5. Are these different? It's hard to really say. Probably not. Those numbers are significantly higher than what is seen in rheumatoid arthritis alone. And Kevin did show here, as he did with tofacitinib, that there's regional issues that in Southeast Asia there's a much higher risk, as high as 6 or 7 events per 100 patient years. It is certainly lower in the United States and in certain places of the world. It may be influenced by steroid use and other comorbidities. So the good news is that these events were fairly simple, straightforward shingles with very few multidermatomal or ocular shingles reports being seen. So that's an important safety advance for a new drug, baricitinib, that's on the cusp of being evaluated by the FDA. Interesting reports about pregnancy appeared. I had a report with Emily Fishman, one of our medical students, who did a review of our patients and showed that it's really not the drug that gives risk to the mother and child. It turns out it's disease activity. It turned out that the patients who had either worsening of their disease or who had unfavorable fetal outcomes were the patients who had moderate to high disease activity. This is a message that's been seen in other studies, including the Corona study we did a few years ago. The work of Megan Klaus shows the same thing. Again, you probably should worry about drugs, but like I wrote earlier, it's not the caboose that kills you. That's a blog that you should look at. It's really the disease activity. It's the engine, and that's what drives infectious risk. There was a nice important study done by Megan, a prospective study of 18 patients, that's Megan Klaus, that is, looking at lactation and whether or not you can breastfeed while you're taking certolizumab pegol. And it was shown in their study that 56% of patients had no detectable certolizumab, so almost 2/3 of patients had none. But there was over 40% of patients actually who very, very low levels of certolizumab were found in breast milk. They did not look at the infant's blood. That was actually discouraged by the FDA in this particular study of 18 mothers and their children, and this included RA patients and IBD patients. But they did find very low levels. On average, the normal level achieved in a human blood is about 15 to 20, I think it's micrograms per ml, and with certolizumab pegol in the breast milk, it was 0.03 or less in many cases. So again, it is found in a minority of individuals, but the amount that's found is almost not important. And I think that from these data, most would say it is safe to breastfeed with using this particular drug. And a very important report about what to do with infections and perioperative management. Actually, there was an ACR guideline issued and a VA report. The VA report from JUO et al. Looked at the VA services and had an analysis of over 9,000 surgeries and over 5,500 RA patients, and looked to see whether they were on or off of methotrexate, Plaquenil, hydroxychloroquine, and a TNF inhibitor. It turns out when you compare those that were off and those that were on, being on any of those drugs did not increase the risk of postoperative infection, did not significantly increase the risk of postoperative wound infections. So important data. We need to see the full data set when it's finally published, but this is evidence that sort of backs up the ACR guidelines that were presented at the meeting. There was a guidelines presented by Susan Goodman and Jazz Singh. These are a combined effort from the ACR and the AAHK. I'm going to guess that's the American Academy of Hip and Knee Orthopedists. And they got input from patients, and they basically had to base their data on a lot of input, not a lot of RCT evidence, but that with what was available, they came up with several things that you can hang your hat on for rheumatoid arthritis. That one, it's okay to continue the use of methotrexate, leflunomide, hydroxychloroquine, and sulfasalazine. That you should withhold biologics only before surgery, and basically hold it for one dosing cycle. You should hold tofacitinib for 7 days prior to surgery. And that when it comes to restarting these drugs, if you do hold them, that you should restart them 14 days after surgery. My advice is always to restart the drug when they're discharged from the hospital, have the sutures out, and there's no evidence of infection. 14 days sounds about right for someone not having any post-op complications. So there are some guidelines that you'll be seeing in the literature probably next year, published in A&R for both RA and for lupus. It's recommended in lupus that you continue immunosuppressives and that you hold them for non-life-threatening disease, but that you continue them in severe disease. But again, you need to look at the data from the report that's coming out next year. A very important study, one of the last studies I want to review, is the OPAL study. This is the use of tofacitinib in psoriatic arthritis patients who have been DMARD failures. So these patients have not yet received a TNF inhibitor. They have psoriatic arthritis, over 400 patients, most of whom were taking background methotrexate. And when you look at the 3-month outcomes, it was shown that tofacitinib was effective. The most effective dose was the 10 milligram BID dose, the dose that's not currently available. And the 5 milligram was almost as effective. 5 milligram was 50, 30, 15, 17 for ACR20, 50, 70, and the 10 milligrams was 60, 40, 20, almost as you'd expect for a DMARD incomplete responder when they used the 10 milligram BID dose. They had very good skin responses in these studies, about 40% at 3 months and 50, 60% at 12 months. But again, the issue is the dose. Is it 5 or 10? In a trial against etanercept in psoriasis, it was only the 10 milligram dose that worked in psoriasis compared to the etanercept response. So it'll be interesting to see how this is viewed by the regulatory agencies and whether this leads to approval of a JAK inhibitor in patients with psoriasis. Anyway, that's it for this week in RheumNow.com. Be sure to go to the website to find these citations and their links. Be sure to go to the website and register. Be sure to go to the website and look at what we covered in the 4 days of the ACR meeting in Washington. I would appreciate your feedback and input as to how we could do that better. Take care. See you next week.